Hello, welcome to VerEvMed Studios, where we are alive in an undisclosed location somewhere in the American Northwest. Today, I'm going to talk to you about parasitic disease caused by schistosoma. We're going to discuss briefly its life cycle, and then we're going to look at a new paper, about a paper that's published about a year ago, discussing a possible avenue for eradicating or at least ameliorating this very deadly and moribund disease. So again, we're VerEvMed, and let's get started. So again, schistosoma. This is a VerEvMed mini lecture. My name is Dr. Daniel J. Guerra, and this is March of 2017. What is VerEvMed? We're a web-based company, and we are scientists verifying published evidence in medical biosciences. So we'd like very much if you would sometime visit our website and take a look at what we do. These lectures are free, and we're trying to give them to you so you get an idea of the kinds of things that we explore and examine in the primary literature. So what do we know about schistosoma? We can take a look at the World Health Organization statistics for 2015. Somewhere close to 220 million people required some kind of preventative treatment for schistosomiasis. Of those, some 66.5, almost 67 million people were reported to have to be treated for the disorder. And of those, upwards of 200,000 deaths have been attributed to schistosoma type diseases. That's on the average per year. What is schistosoma? First of all, there are 5 total species. Major one that we normally, the first one it was named after is called Schistosoma mansonii, but there are 4 other species shown there. They're distributed throughout 3 major continental regions: Africa, Asia, and in South America. So we don't see many cases in the Western Europe or in the United States, except those that have been brought in from overseas. It shows you the life cycle. This is one of those parasites that has an intermediary host. So let's take a look at this life cycle coming from the end of it, where you have paired adult worms down here that migrate to the mesentery venules of your bowel and rectum, and there they lay eggs, and they circulate to the liver. Those eggs can circulate back to the liver, or they can be shed in the stool. When they're shed in the stool, that's completing the life cycle. So let's continue on. So there's S. mansonii and S. japonicum. There's also a schistosoma hematobium. The hematobium is sort of a different character because it's more blood-associated disease. That's why it's called that. But all 3 of them are from the same genus, so we can understand their life cycle by just examining this slide. The eggs then are in fresh water. The eggs hatch and they release a form called myricidia. Okay, that's a stage of the parasite. The myricidia then are the stage that penetrate the intermediary host. In this case, it is a freshwater snail. There are multiple types of these freshwater snails, also known as gastropods. Once in that snail, the schistosoma will go through a sporocystic phase where there'll be successive generations. Ultimately, there will be cercariae that are released by the snail. These cercariae then are going to be free floating in the water, and that's where humans acquire the infection. So you don't need an open wound or anything like that. These come in directly through the hair follicle. So you can wade in the water, such as in a rice paddy or in shallow fresh waters in Africa, Asia, or South America, wherever there are these snails and the potential for them to be carrying the schistosoma. This will then enter your bloodstream via traveling down the hair follicle, following heat gradient. Once it gets into the body, then its cercaria will lose their tails. This is section 7. This is step 7 here, and they will become schistosomuli. The schistosomuli will then circulate in your blood. Eventually, they will migrate to the portal blood and the liver, and they'll mature there into adults. You need both a male and a female, just one of each. Then they mate basically for life, and they can be mated and exist in your mesentery for decades. When they do that, they constantly generate eggs. Some of those eggs make it back up into the liver, and that causes some of the symptomology we'll see here in a moment. But the rest of the eggs then are shed out of the rectum and into the stool, and then they enter the fresh water and they complete the life cycle again through the snail. So you can have repeated infections of schistosoma even within 1 year. And this is the real problem. And there's no built-up immunity to schistosomiasis, primarily because of the way they evade the immune system. So the blood-borne stages evade the immune system. It's believed by coating with serum proteins. These serum proteins can be serum albumin. You can also, a common serum protein are also lipoproteins. The male and the female mate for life, and they can exist again in the mesentery venules for decades. And throughout that period of time, they can shed eggs. And those eggs, some of them are going to make it to the stool, or if it's hematobium, it's going to make it to the urine. And likewise, they're going to be flooding back up into the liver, and they're going to cause a lot of symptomology that you get. So now it's known that there is a schistosomal fibroblast growth factor. So SMFGFR for the Mansonii species. And there are two forms, A and B. Now, those are receptors for the fibroblast growth factor. Now, it's known that those growth factors are necessary. Now, this is for the schistosome, right? This isn't for the snail. This is for the schistosome itself, the parasite. But those two growth factor receptors are necessary for very important aspects of reproduction of this parasite. They're necessary for gonadal development, sexual dimorphism, and the egg-laying process itself. Obviously, then these are going to be targets. These receptors are going to be targets for potential pharmacotherapy. But what happens when you see a presentation of this in the clinic? You get generalized lymphadenopathy, hepatosplenomegaly, which means an enlargement of the liver and the spleen. You can get a rash, fever, right upper quadrant tenderness, urticaria, and of course, bloody stool. When you have chronic schistosomiasis, that you can get these kinds of symptoms. Portal hypertension with abdominal distension, very common presentation. You'll see a picture of that in a moment. Again, enlargement of the liver and spleen, pededema, pallor, distended abdominal veins, and ascites fluid can also be generated from the blockage of the liver. There are CNS symptoms, including focal neurologic findings, seizures, and spinal cord lesions. You can also get renal failure and chronic disease, anemia, and of course hypertension because of that, because of renal failure. There are also some indications of right heart failure, and there are genital lesions, which include ulcerations, hypertrophic lesions, and nodular lesions of the cervix, vulva, or vagina in women, and sometimes these can cause menstrual disorders, cervicitis, and indeed even infertility. This is again a photograph showing you sites of entry of the infectious stage of the schistosoma. And here you're seeing this bloating that occurs because of the blockage of the liver. Now, children may develop anemia, malnutrition, and cognitive disabilities when they are infected with schistosoma. The most common drug that is administered is praziquantel, and that is the primary form of treatment. But again, you can get multiple infections of schistosoma. So that doesn't mean that you're getting cured from it. You have to constantly then take more praziquantel, and eventually this chronic infection can lead to those symptoms and signs, which can increase morbidity, and even that's where mortality comes into play. So in the laboratory, there are direct peristological methods. You can detect the eggs in the urine if it's hematobium, and of course in Mansonii, and in the other species, the eggs are in the stool. And there are multiple methods to detect those eggs. There are also molecular methods, which we don't need to go into. These tend to be the most common. You usually look for the eggs in the stool. There's also a blood examination. Just roughly speaking, you can get some eosinophilia, leukocytosis, and anemia. But of course, these are going to be general features of a parasitic infection. or some other kind of bloodborne infection, and you're still going to have to do that egg diagnosis to be able to really pinpoint that you have schistosoma. This slide shows you basically the lifespan and the kinds of things that go on inside the host. So there is an immune response that can be launched. Again, it's a very poor one because the macrophages need to be able to pick up on the fact that they're invaded by the schistosoma. And the blood stage of the schistosoma are coated with serum proteins. And because of that, macrophages or other professional antigen presenting cells are not often likely to be able to find the schistosoma in the blood and then do that presentation and then induce an acquired immune response, which is, of course, the much more potent one, which will give you memory. So you're getting from the innate immune response to the acquired immune response. When that happens, you will get resistance, and there is some resistance as long as you only get a few infections. So you get a Th1 response and a Th17 response. Those are two different types of T cell lineages. You also later on get a Th2 response. There is inflammation, and there's also fibrosis, and sometimes this can lead to malignancy in the host. There's an allergic response. When you have a robust immune response, you can get some allergic response. You can get some immune complement forming. You can also get amyloidosis. In acute disease, you can get urinary infection, the hepatointestinal disease, cardiopulmonary, and then leading to the chronic disease, which we've already discussed some of those signs and symptoms. I want to show you a little bit about the immune cell lineages, particularly the T cells. So you need this antigen presenting cell here. So this could be the macrophage or a dendritic cell, and that's going to be able to recognize, when it does recognize the schistosoma in the blood, it's going to be able to tear down that parasite, and it's going to present fragments of its protein complement on its surface. When that happens, and you do make an APC, a professional antigen presenting cell, that's a good thing because then you can trigger the T cell response. Again, the T cell response is multiple and varied. The Th17 response involves a rapid inflammatory response, IL-17 particularly in 21, 22, and 23. These are cytokines which are pro-inflammatory. Normally you get a Th17 response from extracellular bacteria, but again, you can also get a response like this from schistosoma. Fungi will cause it, and also a lot of autoimmune diseases turn on the Th17 response. A Th1 response requires these cytokines to trigger the differentiation of the naive T cell after APC presentation. You need those cytokines, and then Th1 will generate these cytokines, again, pro-inflammatory. And what does Th1 do? It's a cell-mediated immunity. It also is involved in inflammation. It involves often intracellular pathogens, and again, autoimmunity and inflammatory responses. The Th2 response is considered more likely for parasites. These are the interleukins or the cytokines that are generated, all pro-inflammatory. These are the cytokines necessary to differentiate into Th2. And again, this is going to be the antibody-mediated immunity. So you're going to have here an interaction with plasma cells. You also get, after these particular signaling molecules, which are also cytokines, the differentiation of naive T cells after APC presentation, the T regulatory cells, which generate these cytokines. T regulatory cells actually cause a tolerance of the immune response, and they tend to tune down the immune response and generate what's known as lymphocyte homeostasis. So they're going to counteract that potent pro-inflammatory response of Th17, Th1, and Th2. Okay, so it's a little, very small primer of the acquired immune response, which we would like to see in something like a schistosoma infection, but which is not very robust, again, because the schistosoma, the blood-borne stage of it, can often evade the immune response unless it becomes chronic. So we gave you that very brief introduction of schistosoma so that we could talk about this one paper. So let's take a look at the title of this paper. We're looking at a novel Toll-like receptor. Now, TLRs are part of the innate immune response, not the acquired immune response. So these are receptors that are going to be on the surface of things like antigen-presenting cells, like macrophages and dendritic cells. And once they process that invading parasite, then they can turn on the acquired immune response. But the first stage is for them to then bind to and interact with the Toll-like receptor. Now, notice we're not talking about the Toll-like receptor in the human host. We're talking about the Toll-like receptor in the intermediary host, in that gastropod. Okay? So what the idea is if we can understand the immune response in the gastropod, in the snail, maybe we're able to make the snails resistant to infection by the schistosoma, and in so doing, decrease the amount of snail load or bearing load of the schistosoma, therefore decrease the amount of schistosomes in the freshwater system, and if not eradicate, greatly reduce the reproductive cycle of the schistosoma. So again, that's what we're looking at here. So let me just briefly remind you that praziquantel will kill Schistosoma mansonii and the other species, but you often get multiple infections in the tropics, like in Africa and Asia and South America, those regions. And it's particularly devastating in small children who get those multiple infections. And cumulatively, it can cause severe morbidity and mortality, including central nervous system issues and death. So the idea is to disrupt that life cycle. So that's one of the ideas. Again, we can go directly at the pathogen, that is this particular parasite. We can also look at host, that is human immune response. So there are drugs targeting all three of those areas. But right now, I'm just talking about this paper published in PLOS Pathogens about a year ago. So what about looking at this intermediary host? So perhaps we can see whether or not this Toll-like receptor has any specificity when it interacts with the schistosoma. Okay, so is there a Toll-like receptor, and what's the role of it in cellular, that is innate immunity in the snail when it's being invaded by the schistosoma? So S. mansoni resistant strains of the snail displayed higher levels of a specific Toll-like receptor compared to the susceptible strains. So it looks like jacking up the expression of that particular Toll-like receptor makes the snail resistant to the schistosomal infection. So what else can we say about this? What else do we know about this? Well, from this paper, it was shown that the transcript level, okay, that is the RNA that's made from, which is going to eventually turn into the protein for this Toll-like receptor, in snails, that transcription occurs when they are challenged. And you get a tremendous increase in the transcription of that receptor, of the Toll-like receptor, which again is going to give you resistance, up to about a 27-fold increase in expression. So that's a very robust interaction, right? Now, the expression of that Toll-like receptor in the susceptible cell lines was not significantly increased. So the resistant snails, okay, the resistant snails are going to make a lot of that Toll-like receptor, and that's going to block, right, or it's going to induce an innate immune response. So it's going to stop the infection from occurring and becoming full blown. But in susceptible snails, you don't get that robust induction of the Toll-like receptor. Indeed, this paper describes a knockdown using siRNA, okay, so that's interfering RNA. And this is basically when you make interfering RNA, you get sequence homology to the messenger RNA that's normally the target for what you're asking is associated with this specific research agenda. And this research agenda is asking whether or not the transcript level of that Toll-like receptor is mediating this resistance. So if you use a small interfering RNA and you knock down that transcript, what happens, okay? Well, using that, or in fact using antibodies, okay, just regular antibodies to that Toll-like receptor, you get a significant alteration of the resistant phenotype. And that will yield patent infections of almost 50% of those resistant snails that are naturally resistant. This is a proof of concept. This is saying that you get this huge increase in this Toll-like receptor expression in resistant snails. And if you block that induction by using either an antibody to that receptor, that's a protein level blockage, or you block it by small interfering RNA, you get back to looking like a susceptible snail. Okay, so that's what this experiment was done for. So this Toll-like receptor looks like it is a key immune receptor that is capable of influencing infection in when you challenge these snails with Schistosoma mansonii. So perhaps this will be a new strategy for the eradication. And how would we do this? Well, one idea, and there are more than one idea here, but one that we may throw out there is, can we target that toll-like receptor? And there are a couple of ways of doing it. Maybe we can use an agonist to activate that toll-like receptor, or maybe we can use some other kind of analog, maybe some kind of antigen from the schistosoma that will superinduce the expression of that toll-like receptor, therefore preventing infection of the snail, therefore knocking out the intermediary host, and therefore disrupting the life cycle of the schistosoma. So again, this is one avenue. There are two other ones, again, going directly for the schistosoma, finding drugs that will target the parasite itself, and also drugs that which will affect in the human. Of course, we already have praziquantel, but maybe there are drugs that we can use in human to help jack up that immune response in humans as well. And there are problems with all three of these strategies, but I like this one because it goes after the intermediary host without needing to go to the human host. So here's our email address. Please send any interesting email our way. All email, even if it's not particularly interesting, it'll probably be interesting to us. There's our website, www.VerEvMed. Again, we are a company where scientists verify published evidence in medical biosciences. And we very much like to hear from you, and thank you very much for your attention. Again, this is the VerEvMed Studios. We're coming to you from an undisclosed area in the American Northwest, high in the mountains where we're experiencing a lot of torrential rain. Thanks a lot again for your attention. Ciao.